Okay, this might be the first recorded video of the unit. We're gonna kind of see how this shakes out. But DNA replication, important topic. Let's just jump right in because I'm almost out of time. So, first thing to know about DNA replication is that it is what we call a semi-conservative model of replication. And so, I think that's most easily understood by looking at the other possibilities. So the first is a conservative model, where basically the template strand that we'll call the parent strand gets completely conserved all throughout all possible replication. So when I make a copy and I have this double helix of new DNA that I just made, that new double helix molecule is entirely new. And so we could track the initial two strands of DNA and they would stay together no matter how many times they replicated. A dispersive model is the other extreme, where basically we don't care that they're individual strands at all. They just get chopped up, copied, mixed and matched as we replicate. And so the initial parent strands are broken up and spread throughout both daughter strands and continue to spread out and fracture as we go through. What we actually have is semi-conservative, which is just kind of a mixture of the two. And so whenever I copy my DNA, those two strands get separated, and the new daughter DNA double helixes will each have one half of the helix be from the initial parent cell. The other half of the helix is new and just freshly copied. So that is the model that exists, is semi-conservative. The strand split, and each daughter gets one new strand, one old, which should help spread out errors and things like mutations so that they don't compound. We know this because of one more experiment called the Mendelssohn-Stahl experiment. They used radio labeling again on the parent DNA only, and then let them replicate and copy that DNA with non-radioactive elements. And basically when they did that, the radioactivity stayed in single strands all the way through, and so most of the radioactive nitrogen, in fact, all of it was just in the initial two strands. So how does DNA replication actually work? We are going to go over this in class. It is confusing. I want to give you a primer on it, before we start. So you have a few proteins to know. Am I going to totally get... Oh, no, we're kind of good. Proteins to know. First, DNA helicase. Looks like a zipper, and that's what it is. It unzips your genes. There's a pickup line there if you work hard enough. So DNA helicase unzips the DNA because when the DNA is in its double strand form and stuck together, we can't get any proteins in there to make copies of it. The next one to know is topoisomerase. This one, a lot of people get it, forget what it is. It is correcting of overwinding. So basically when you're unzipping this and doing all this manufacturing of new DNA strands, topoisomerase stops the DNA from like twisting and getting tangled in itself. Kind of like when you have like now no one has corded headphones anymore, but when you used to have corded headphones, you would put them in your pocket and the cord would get all mixed up, and it would be a pain to untangle it. Topoisomerase basically just says, I'll just cut the knot and then untie it once it's cut, and then I'll join them back together. So it just relieves what's called supercoiling, relieves tension. Polymerase, polymer, ACE, it is the protein that makes polymers, in this case of DNA. So it's just going to be adding the DNA to the RNA primer, which it requires before it can work. It can't add nucleotides onto nothing, so it has to have a little bit of, in this case, RNA there to actually start the process. And then lastly, we have ligase, which is going to actually just glue the the DNA strands back together in a particular form that we'll talk about later. So when we have DNA replication, we get these things called origins of replication, where because we have these two linear chromosomes, you'll get multiple what are called also called replication forks opening up at various points along the chromosome so that it can all happen quickly and efficiently. So you won't just have this starting at one particular location. It will start in multiple locations and spread until they meet each other. because we have such large chromosomes, it would take forever if we had to only use one protein basically in one origin to go through everything. So what is actually happening? As I said before, DNA synthesis has to start with a primer. It is a strand of RNA. It's very short. It is complementary to the DNA sequence, and so it is sticky with that DNA sequence. And it just provides a spot for polymerase to come in, attach, and then begin to add nucleotides. Now, we have a rule that DNA polymerase can only add nucleotides in the 5' to 3' direction. And that produces some weirdness because we have two strands, but they're going opposite directions. So when we have a replication bubble opening up, you get two different things that happen. One of them is simple. It's called the leading strand. And because that one is already going in the 5' to 3' direction, see how this is 5' up here in the lighter color, it's growing towards the 3' direction. You can imagine topoisomerase in here, unzipping further, going to the left. And so the direction of replication is in the same direction as DNA helicase. And so nothing fancy needs to happen. We can continue growing that continuously. The other strand is different because DNA is anti-parallel and they're flipped relative to each other. This strand, growing 5' to 3' is going to the right, but the overall direction of replication is going to the left. And so on one of the two strands, you will have DNA polymerase working opposite the overall direction of replication, meaning you can't synthesize it continuously. You need ultimately many different primers, and it's going to be done in many different chunks. So essentially, what happens is that the lagging strand will be synthesized in small chunks. It'll go out a bit, primer comes in, DNA polymerase makes a short strand, DNA ligase glues them together, opens up a little more, DNA polymerase goes in, makes a short strand, glues them together. And again, this all results because of anti-parallel elongation. DNA polymerase can only add nucleotides to a 3' end of a growing DNA strand, so this end here, because that is what is accessible. And so DNA can only grow in the 5' to 3' direction, which for one of the two strands means it's going to go backwards. So we call them the leading and the lagging strands. The leading strand goes basically is DNA polymerase going in the direction of replication. Lagging strand is DNA polymerase is going opposite the overall direction of replication. So you get these things, you get the small fragments that are called Okazaki fragments. They are just these small fragments of DNA. It's basically how many short DNA can you make before you run into where you started. And so you can see up here, it's like you start at 1. So we start at 1, make the DNA, then DNA helicase has unzipped more of the DNA, so I can get a new RNA primer in. DNA polymerase comes in again, makes another Okazaki fragment. DNA ligase glues them together. And so you get this pattern that you can see here in the top right. You will be asked to explain this. You'll see this. I would recommend drawing this light yellow box up in your notes. Because you can see at the origin of replication, leading strand on top grows continuously, lagging strand grows discontinuously on the bottom, and then it is flipped on the opposite side of the origin of replication. The last thing I want to talk about briefly is polymerase chain reaction, also known as PCR. You're not really going to have to know much about this. They'll have to give you background. I just wanted to explain it briefly before you see it on a test or something. So you've probably seen this with like COVID testing or things like that. It's used in diagnostic testing, so seeing if you have a virus or something. It's used in research. It's used in DNA sequencing like 23andMe. It's used in vaccine manufacturing. It's used everywhere. The essential idea is that you are just making DNA replication happen in a very specific way. So you need whatever DNA sample you want to be amplified. You need some nucleotides to build new DNA strands. You need a buffer solution to hold everything in. You need primers and RNA primase to actually start it. And then the last thing you need is Taq polymerase, which is a DNA polymerase that is isolated from a thermophilic bacteria. It lives in some hot springs, and so it's active at really high temperatures. And so the main idea is just that you heat these things up. That separates the two strands. You let it cool down a bit, let the primers attach, and then you heat it up again and allow Taq polymerase to come in and actually make the new DNA strands. The result is you just get a ton of copies of the DNA strand that you put in, which is a very useful thing for research. And that is this that video. I screwed this up. That's all. Farewell. Short, hopefully not too bad. We'll talk about it next time too. Bye.